Hello, I'm Sydney Warfel, and I did my toxicology presentation on the toxin aconite, a lethal poison that you may be able to find more easily than you think. You might even be able to find it in your backyard or the forest nearby your house. Aconite comes from the flowers in the genus Aconitum. The flowers in this genus are plentiful. Over 250 different species exist, but most are extremely toxic. These flowers are described as brightly colored, ranging from light blue or purple to bright yellow. Due to the low maintenance as well as the bright and attractive colors, this flower is commonly grown in home gardens even though it is highly toxic to both humans and pets. These flowers are able to live in unfavorable conditions and will bloom so long as they are exposed to sunlight, making it a perfect addition and easy plant to keep alive in gardens that are easily forgotten about. The issue, however, with these plants being grown in home gardens is that they are borderline invasive. The flowers are rapidly reproducing and grow in colonies that can range from just a few plants to up to 20 plants per colony, and one plant can parent many colonies per season. The main species that contributes to the toxicology field is Aconitum nipalis, which is also known by a variety of names such as wolfsbane, monk's hood, and the Venus's chariot. These dark blue or purple flowers can bloom mainly in the cooler months, but can bloom anywhere from June to October. They can grow from just 1 foot to 3 feet in height, but height varies from plant to plant. These plants also possess inflorescence in their flowering, meaning that the flowers grow in clusters around the stem and are called some of the most attractive flowers that grow in the U.S. These plants have been found in places all over the world, but are native to Asia and North America due to the temperate weather and common sun exposure, being ideal for the growth. The plants have spread to other areas due to the attractive flowers drawing the attention of gardeners that plant them in their homes or in gardens, but the rapid reproduction has caused them to spread to the outside environment where they are less likely to be managed and kept contained to one area. In regard to toxicology, the poison that is possessed by this plant is known as the queen of poisons, from the name that was given to it by the Greeks because of its extremely deadly properties. The poison itself is drawn from mainly the root of the plant. However, the entire plant, including the flower, can possess the toxin. The symptoms are very painful and can occur anywhere from a few minutes after ingestion to 5 hours after ingestion. These symptoms target the heart and central nervous system, causing a numbing feeling, abnormal heart rate or rhythm, and gastrointestinal issues such as vomiting, nausea, and abdominal pain. It is said that if someone survives the first 5 hours, they are likely to live. Those who aren't fine will likely die from half an hour to 5 hours after the poisoning occurs, and the most common cause of death is paralysis of the respiratory system, stopping the heart or causing the lungs to refuse to take in air. When talking about where this poison came from, there are many historical theories, but the most common is from Ovid's Metamorphoses, a series of poems written in Rome about 8 CE. Ovid explains that the poison came from the mouth of Cerberus, and when the three-headed dog's drool dropped onto the ground, wolfsbane grew plentiful. It is now thought that they thought this because the poisoning that occurred via this toxin had similar symptoms to that of rabies, and the Romans easily associated the two. In other parts of mythology, the poison had been relevant for more morbid reasons. Athena used wolfsbane flower to transform Arachne into a spider, and Medea used it to try and kill Theseus. In history, the poison was used more practically by hunters and gatherers to dip their arrows in to create poisoned arrows. These arrows were then used to kill large predators, such as wolves, that were considered a nuisance, but they were also mixed into poison baits to kill these predators as well. This is likely where the nickname wolfsbane came from. It wasn't until 1881, when Dr. George Henry Lamson was found guilty of poisoning his brother-in-law, that aconite was known for being used for murderous purposes. He wanted to be involved in the family trust fund of €3,000, which would go to the brother-in-law, Percy, after he came of age. George killed Percy with aconite-laced cake as well as an aconite pill, which ultimately led to police questioning him. While he wanted the money, he was a horrible liar and confessed to his guilt and was hanged for murder in April of 1882. While most uses for aconite are less than moral, the Department of Traditional Chinese Medicine came up with an experiment that could test the poisoning effect on tumors rather than people as a whole. The scientists took the tumors in mice and injected them with a varying amount of aconite, ranging from none at all to doses as high as half a gram per milliliter. The dynamics of the body mass of the mice were recorded on a daily basis for 3 consecutive weeks before the mice were euthanized if they had not already died from natural causes. The survival times of the mice were recorded, as well as the size of the tumors after death. Shown in the photo on this slide, the mice who were significantly exposed to aconite had smaller tumors when compared to those that were exposed to none. The middle two fall somewhere within the middle, but the tumors were getting smaller with the higher dosages of the toxin. After the experiment, the results were gathered and placed into multiple visual aids, such as this graph pictured as well as the image on the last slide. This graph shows that the volume of the tumors decreased even more with higher doses of aconite, putting the last photo into another comparison. The model mice had the largest tumor volume all around, while the high doses had the lowest tumor volume, being nearly half, if not more than half, the volume of the model mice. In addition to the sizes of the tumors going down, the quality and activity of natural killer cells in the mice exposed to aconite significantly increased. These cells work within the immune system to seek out and destroy cancerous cells to keep them from spreading and therefore strengthening the adaptive tumor immunity. The study did not find out that aconite had a significant effect on the number of B cells, though. Other results showed that the mice exposed to aconite had an increased chance of survivability. While there was a drop in the beginning, the days following the administration kept within a steady rate that had better survival percentage when compared to the other dosages. The model rats had less than 50% remaining in around 26 to 27 days, while the mice exposed to high doses of aconite did not reach 50% until around days 31 to 33. At the end of the experiment, all of the model mice had died around 33 days, while the mice with the high doses survived until days 38 to 40. While the days are not largely separated, the improvement is still noticeable enough to be noted. Aconite also seemed to increase apoptosis, or programmed cell death, in the tumor cells more rapidly than untreated tumor cells. Killing off the cells gives a smaller window of time for each of the cells to duplicate and create a larger tumor. On top of the apoptosis boosting, aconite also seemed to inhibit the activation of T cells in the body, which act as cancer blockers. How is this information useful for us? The experiment may have been done on mice, but it provides valuable information that could help us in the future. Experimenting on ways to shrink these tumors or keep the tumor cells from duplicating can help scientists or medical professionals find more ways to treat tumors in humans. This will help us work to restrict or shrink the cells in us as well as in other animals. The process of adding this toxin to tumors also helps us see exactly how the body works in the process of getting rid of said tumors. If we can see exactly what kind of cells needed in order to target a tumor, we may be able to find other ways to help the body produce them. If it is discovered that we can use aconite in the human body without causing severe side effects, this would also help the name of the poison from becoming one that has no other use than to cause harm. These are the sources.